All right. Well, here is our lecture for Monday, and yeah, we're going to discuss basically the differences between micro and macroevolution, and we'll talk a little bit about speciation. We'll talk about how modern understanding of genetics is complicating this issue a bit, and then finally we'll talk about how does an interventionist perspective help shape our understanding. So first question is, what is microevolution? And so microevolution is really it's a change in allele frequencies. So usually what we're and this is at the population level. Sorry, a change in allele frequencies is at the population level. So when we're talking about microevolution, we oftentimes represent it using what's called Hardy-Weinberg equilibrium. And so if you look at the top of this slide, you see P plus Q is equal to 1. This is setting up a relatively simple illustration in which we have two different alleles for a particular gene. And so the frequency of P, which we usually use to represent the dominant allele, plus the frequency of Q have to equal 1 because there are only two alleles. And so the proportion of each allele, or the proportion of both alleles combined, have to equal 100% of the total alleles. And then the next equation represents P squared is the proportion of individuals that are homozygous dominant. 2PQ is the proportion of individuals that are heterozygous, and Q squared is the proportion of individuals that are homozygous recessive. And if the population is not currently experiencing microevolution, then all of those should match up. Your proportion of individuals that are homozygous dominant should be P squared, and your proportion of individuals that are heterozygous should be 2 times P times Q. If any of these don't match up, then it's an indication that the population is experiencing microevolution, that the frequencies of the alleles are changing in that population, and that's what microevolution is. So microevolution is fueled by seemingly probably the most powerful one of these is genetic drift, random genetic changes. Those are going to be especially powerful in small populations. Microevolution can also be fueled by gene flow, either individuals leaving the population or entering the population. Microevolution can obviously be fueled by mutations. If you are generating new alleles, you are shifting the allele frequencies. And even if you're generating the same mutation or the same allele over and over again, you're still changing from one allele to another and changing the allele frequencies. Natural selection, this is probably the most famous of the microevolutionary forces, although there's quite a bit of debate about how powerful this force actually is. But this is the idea: if one of these alleles is favored, that is, an individual with that allele has increased survival or reproductive capacity, then the allele frequencies in that population are going to shift towards whatever that successful allele is. And then sexual selection, this is really if they have increased reproductive output because their allele makes them more attractive to the opposite sex. So these are our mechanisms of microevolution. There are some others, but these are the big contributors to microevolution. So speciation, formation of a new species, this happens as a result of microevolution. And this isn't just some abstract extrapolation of microevolution. We have actually witnessed this happen in action. We're watching it. A few days ago, we talked about this happening in orcas, and then we've seen through records of this happening in Galapagos finches and other animals where we've seen these kind of radiation events of new species forming through a process of microevolution. So then we have a question of what is macroevolution. And so there are two different ways to think about this. One is any evolution above the species level. And that is one way of thinking about macroevolution. I don't think it's a particularly helpful way of thinking about macroevolution, but it is a way. The other one is that it's major phenotypic changes resulting in higher taxa. And so if you look right here, here's the idea there. Microevolution and speciation, and maybe even generating new genera, although we'll talk about that in a later lecture, would be under this category. And then macroevolution is going to be major phenotypic changes resulting in higher taxa, like the split of a common ancestor to generate both mammals and reptiles. I think that's a much better way of thinking about what macroevolution is and the specific meaning of that, what's conveyed typically when you hear macroevolution. So what is required for microevolution? How do we set a population up so that it can fluctuate? So for the most part, microevolution requires excess individuals produced. Now, there are some exceptions to this. You could have microevolution fueled by genetic drift without producing more individuals than can possibly survive. But in order for natural selection or for sexual selection to work, you have to produce excess individuals. You have to produce more individuals than can survive to reproduce. You also have to have variation, and this is an important one. And you know, one of our mechanisms of microevolution is mutations, but we have to have some variation in order for the allele frequencies to change. You can't have just one allele of a particular gene. You have to have two or more, and because of that, you're going to have some variation. Not all of your individuals are going to match. The book actually makes an illustration of if every offspring of a breeding pair of mice survived to make that number of offspring again, that within just a short matter of time you would have a pile of mice with 17,000 miles thick. And so obviously the indication is there that a lot more individuals are produced than even survive, but within that pile or within the actual realistic population, you have quite a bit of variation in those forms. And then especially for natural selection, some variants have to survive better than others. So really this question is being answered as what is required for microevolution by natural selection. But for microevolution by genetic drift, really not much is required at all. Genetic drift is random genetic changes, usually as a result of small populations, but not always. You could have a massive natural disaster, kill a number of individuals in the population, and leave you with a potentially very different proportion of alleles than what you had previously. So how does speciation happen? How do we actually generate speciation through processes of microevolution? There are people that would suggest and argue that you cannot have speciation without geographical isolation. That geographical isolation is an absolute must in order to generate a speciation event. Although it would be hard to hold that for all conditions, I would suggest that the norm for speciation to happen is that you start with geographical isolation, and then you develop reproductive isolation. But it seems to be that there are other isolating mechanisms. It's no doubt that you have to develop some mechanism to get the reproductive isolation, but that mechanism may be behavioral, and I think that's what we're seeing in our orcas, where we're starting to develop a new potential species of orca that hunt whales, and so their behavioral differences actually keep them from reproducing together. The use of echolocation versus the non-use of echolocation. You might also develop mechanical isolation, in which the organisms cannot physically reproduce. This would, if it's not, if you don't have geographical isolation happening previous to this, it would most likely be due to drift, some random, very dramatic difference, and then molecular isolation. This again would probably be due to drift if geographical isolation did not happened before this molecular isolation, some kind of a drift event in a small subset of the population, for some reason. Maybe they weren't geographically isolated, but they were kind of at the outskirts of the population and experienced some kind of a massive change in their genetic information, maybe a massive loss of individuals and genetic potential. So next question is, how does our modern understanding of molecular biology shape our understanding of how microevolution and speciation work? And really it's explained or described in this question. How do epigenetics complicate this picture? And really they do. And so epigenetics really fall into one of two categories. One, into the category of regulatory genes, which this is the control of the DNA beyond just the sequence of DNA. So we have a number of regulatory genes. In this picture, these are Hox genes that actually control the transcription of genes. And so you'll see different areas on this fly's body where you have different Hox genes, these regulatory genes being expressed. And so it doesn't matter that you have all the same genes in the cells here as you do here, because not all of those genes are going to be expressed equally in those different body parts, because the expression of those genes is actually controlled by these Hox genes, or these regulatory genes. And so this complicates the picture quite a bit, in that it doesn't matter if you develop, a small mutation, if the regulation of that gene inhibits that mutation from actually being expressed. And so you'll find this even beyond Hox genes, that will have some ways of maybe shutting down one of the copies of those genes. And so even though you may have a novel mutation, it may have no phenotypic impact. Therefore, natural selection can't act on it, because that gene may have been actually shut down. And here's another area there, not regulatory genes shutting this down, but actually regulatory chemistry shutting down. So what we find is there are control mechanisms, regulatory elements that will methylate chromosomes or will acetylate chromosomes. And so when it methylates chromosomes, it condenses them down and makes it impossible for the transcription machinery to gain access to that. And when you acetylate it, it removes the methylation, opens it up for that transcription machinery. So these aren't genes necessarily that are controlling the expression of other genes, but these are methylation and acetylation patterns. And what's interesting is these change through the life of an individual, but can actually be passed down. So your methylation pattern can change throughout your life, and then you'll tend to pass your methylation pattern off to your cells, including your gametes, so that your offspring will inherit your methylation patterns. Your offspring will potentially inherit how you quiet or activate particular genes. And this is really interesting, because this doesn't sound Darwinian at all. This sounds more like Lamarck's ideas, that an organism throughout its life actually develops new features that it needs for its environment, and then passes those off to the offspring. It also doesn't sound random, like this random variation, as your methylation pattern changes throughout your life based on your experiences, your environment, your diet. What that's going to do is it's going to shape your ability to meet the challenges of your environment without any genetic differences. It's just you're now expressing different suites of genes. And so this complicates the picture quite a bit, in that regulatory genes and these methylation acetylation patterns can completely mask new mutations that are there but aren't expressed, and they can lead to phenotypic changes that have nothing to do with a difference in the genome. The genome is exactly the same, and yet the phenotype is very different because of how those genes are actually expressed. So how extensive are the epigenetic effects? Is this something that it's just in theory, it seems like it could complicate it, or is it really complicating this altogether? So here we have 3 different species of salamander. represented here. Here in species A, you have what's called a pedomorphic salamander, that it'll reach sexual maturity without leaving the larval form. Here you have what would be thought of as a more typical salamander as a larva. It is aquatic, has gills, moves predominantly by swimming, and then it goes through a metamorphosis into adulthood, loses the gills, breathes with lungs, or maybe just buccal breathing, breathing along the lining of the mouth. And here we have a third species that has no larval state, or has no aquatic larval stage. It has direct development. This is showing you the egg. In the egg, you have basically this aquatic larva in the egg, but once it hatches, it's just a sexually immature version of the adult. And so we have three different species represented here, and you would imagine that there are significant genetic differences among these three species. But what you actually find is this is not a genetic difference controlling these different life histories. It is a epigenetic difference, that they all have very similar genetics, but the expression of the genes that they have are what influence whether they stay with a larval form. They are pedomorphic and reach sexual maturity in the larval form. They have a more typical salamander life history, or they pass through the larval form in the egg and are born as a small version of the adult. Again, very little genetic differences. It's almost entirely epigenetic. Here's a representation of the lac operon. The lac operon is a suite of genes in bacteria that enable them to metabolize lactose. And so there's a gene for bringing lactose into the cell, there's a gene for metabolizing lactose, and then there's a gene for shutting down, well, we don't know exactly what the gene does, but we think it may shut down the metabolism of other sugars. And so all three of these genes need to be activated in order for the organisms to metabolize lactose. But the genes are there whether lactose is present or not, but the genes are only expressed in the presence of lactose. You have a way of controlling that. When I say you, the bacteria have a way of controlling the expression of those genes. And so the environment dictates what proteins the bacteria makes, but the bacteria are already prepared to metabolize lactose whether or not they ever are exposed to lactose during their life. Very interesting that it seems that these epigenetic effects are very extensive, and in many cases do a better job of explaining the differences among organisms, or the differences in the development and life history of an organism, than changes in the genome. So our modern understanding of molecular biology is complicating the picture of natural selection and random mutations fueling biological evolution. It seems that it's more complicated than that because, again, epigenetics can mask new mutations. So you can have a novel allele that never even gets expressed, so it doesn't impact the phenotype. And on the flip side, you can change the phenotype with no genetic difference. And so now it's making this picture of random mutations and natural selection seeming a little bit more complicated. So how can an interventionist view help? How can interventionists help? First of all, I think it reminds us that natural selection is really not a great mechanism for driving evolutionary change, but it is a great mechanism for selecting against the extremes. And so here's an image of stabilizing selection. And this is more of what you would predict based on an interventionist idea of natural selection. That natural selection is a force to weed out bad mutations, but really is not a great force for generating new and novel forms.
All right, sorry about the awkwardness of the video up to this point. But yeah, picking up, how can an interventionism, or how can interventionism help, or an interventionist view help, is again, it's a good reminder that natural selection is primarily going to be a preserving force, a force that weeds out mutations that are able to be expressed even with the epigenetic factors and are deleterious. Another way interventionism helps is by giving us a better explanation for these phylogenies. And so here we have two phylogenies of a type of parrot, a family of parrots. And in the one on the left, you have the proposed phylogeny. We're starting at the bottom. You have simpler parrots, and as you work your way up the phylogeny to species 9, you're increasing complexity of the behavioral applications of these parrots. So as you move up, you're getting brighter colors, and on top of that, you're getting more complex behaviors. And so this tends to be how you see phylogenies. The assumption is that the ancestor is going to be a more basic form, and as speciation happens, it's going to happen in a lot of ways by accumulation of new features. The phylogeny on the right is basically the same phylogeny, but presented backwards, assuming that the most brightly colored and those with the most complex behaviors are actually the ancestors of the duller and less complex. And so I think an interventionist view assumes that at the beginning these created groups are going to have a significant amount of diversity possible, and as they go through this process of diversification and they specialize, they start to lose some of these complex features that were available, were at their disposal to employ, and as they specialize, they lose some of those features. Another way I think interventionism helps is in explaining antibiotic resistance. So here you have a cartoon of a bacterium chasing an antibiotic. And what's interesting is antibiotic resistance is probably one of the best examples of microevolution that we have. Populations of bacteria can very quickly shift in the direction, allele frequencies in those populations can very quickly shift in the direction of the allele for antibiotic resistance. But what you tend to find when that happens and you study this allele, what you tend to find is that the allele is actually a loss of some typical function, some typical mechanism within the bacterium, and that loss of function inhibits the antibiotics' ability to fight that individual bacterium, because the antibiotic targets the mechanism that's missing in that bacterium. And so antibiotic resistance, while a great example of microevolution, almost always happens as a result of a loss of a mechanism. And this is exactly what you would propose or you would predict would happen from an interventionist view. Another way interventionism helps is it, I think, provides a better explanation for how quickly microevolution and speciation can happen. And so here's what you would predict from a typical view of evolution fueled by evolution and natural selection. You would assume that you have this original ancestor, and then very slow accumulation of mutations, a very slow process of natural selection, leading to microevolutionary changes, geographic isolation, and other isolation mechanisms developing or facilitating reproductive isolation. generating new species, and this is what you would predict under, a phylogeny or an evolutionary history governed by random mutations and natural selection. But this is what you actually find. And so here we have the organisms represented by fossils. And what you find is that actually the greatest amount of diversity of animal, plant, and of phyla actually is very early in the fossil record, and the diversity is all present basically at the same time with no connections. And so if we had to root this back to an ancestor, this evolutionary change would need to happen very rapidly. Well, an interventionist view actually would predict that the rate of speciation it used to be much quicker than it is now. And so here's a proposed idea over how quickly the evolutionary rate. This is what actually matches the fossil record better. Is that there at one point was a rapid speciation rate that has now slowed as you move through the fossil record. And here's the selection experiments in the lab and experiments in the wild. Actually have incredibly high evolutionary rates, and what you see is you have some points in the fossil record that actually have evolutionary rates that approach what we've seen in the lab and what we've seen in experiments in the wild. And it's very difficult to explain this through random mutations and natural selection being the main driving forces of this speciation. But in an interventionist perspective, if God created these different groups of life, these different kinds of life, and then they diversified into various species, we in the text you see some of those, some of these groups, have a wide variety of species. You'd expect that this rate of evolution actually could happen pretty quickly. And so we have one last question to deal with from this chapter, and it's basically would a global flood and a Earth following a global flood actually enable this rapid diversification that you have to have if you have a young Earth, and what you would predict from an interventionist view? Well, the answer is yes, and this is why there would be many unoccupied niches. And so, with a number of places for organisms to move into, you provide a great deal of opportunity for geographical isolation as a subset of a kind enters into a new ecological niche. On top of this, population dynamics would be very unstable as you have very immature ecosystems that are rapidly changing. Those unstable environments actually would provide enormous opportunity for organisms to have very high rates of microevolution as their environment changes so rapidly. And then keeping in mind that natural selection is not the only force of microevolutionary change, that if you have rapid change due to natural selection and you have small populations, you have a great potential for drift to actually increase the amount of microevolution you're getting from natural selection. Rapid geological and environmental changes that would favor allopatry, and allopatry is speciation in separated populations. So rapid geological and environmental changes would actually probably favor geographical isolation, sorry. And then finally, population bottlenecks. So when you take an entire created kind and reduce it to two individuals, you're going to lose a significant amount of the diversity within that group. As a result, you get a bottleneck, your your created kind, the allele frequencies in there are going to change very rapidly, and following that, as you regrow that population, you have a great potential with a small population size for random genetic changes. And so, as you listen to this and you prepare for Monday, keep in mind that we are going to, on Monday, spend some time talking about microevolution and macroevolution and speciation and is macroevolution just a natural and almost required extrapolation of microevolution?